What inspired you to be interested in the Floppy Baby Syndrome?
So what interested me about Floppy Baby Syndrome was that the person I was working with at the time and still now is Professor Nigel Lang, and he'd been working on Floppy Baby Syndrome for many years before I started working for him. So he was able to tell me about the disease and how it affected patients and their families. And I worked out it was a really worthwhile disease to be working on, hopefully that we could make a difference for the families.
What was your process in discovering that actin 1 had a connection with this disease?
So when we're trying to find out what genes cause a disease, is we obviously have to have a look at the DNA from the families, and if there's an affected person or more than one affected person in a family, look at their DNA in particular, and then compare it to other people in the family that might not have the disease. So when we found the ACTA1 mutations or gene defects, we were working with 3 different families, in fact 3 children from 3 separate families who all had this devastating disease, and 2 of them had already passed away. They were all from Europe, and we were able to get DNA sent to us here in Perth, and we were able to analyse their DNA. And at that time, we had a good guess as to what the defective gene might be. We were able to use the information from a colleague, a very well-respected neuropathologist called Professor Hans Goebel, who looked at the muscle biopsies from these children down the microscope. And he saw a particular pathology or particular thing that had gone wrong with the muscle that indicated that it might have been extra protein in that muscle called actin protein. So we thought a very good guess as to what the gene might be that was causing the disease was the gene that creates the actin protein. So my job at the time was to analyse the DNA, the genetic code for that actin gene, to see if we could find any defects in that gene. And that's what we did, and did find that there were defects in that gene. And then after that, we were able to go through the DNA bank that we had here in Perth from different floppy baby patients from around the world, and then look at their DNA and see whether any of them also had defects in the same gene.
What is the screening method and how did you come up with it?
So the screening method that we used was using the what is probably still the gold standard screening technique for DNA, looking at the DNA code, and that's called Sanger sequencing. So it's where we can accurately look at each letter of the DNA code for that particular part of the DNA code that we're looking at. So the actin gene has 6 bits to it, or 6 exons, which are the parts of the gene that get made into RNA, into protein. So we guessed that the disease causing defect or the mutation would be in one of those 6 bits. So we use this Sanger sequencing to hone in on those 6 bits and to look at every single letter in the code in those 6 bits, or the 6 exons. And people still use that technique today to look through the DNA code. But what we also have available these days is what they call next generation sequencing. So rather than us look at a bit of a gene at a time, people can now look at many genes, hundreds of thousands of genes, all at the same time, and look at all the bits of those genes at the same time. So we now have, for example, gene panels where they might have 300 or 400 genes that people know cause a particular type of disease, such as a muscle disease, and floppy baby disease is one of those. So any patient with a muscle disease could be tested for all those 300 or 400 genes at the same time. And one of those genes on one of these panels that we've been using is the actin gene. So currently, patients can be screened that way. But in other parts of the world, and for different companies and laboratories in different hospitals, for example, they can still look at the actin gene bit by bit and see if there's an error in it.
Where do you get your funding from for your projects?
So funding for medical research can be hard to get, and medical research does cost a lot of money. So we have to be very aware of thinking of where we're going to get the next research grant from. A lot of funding in Australia comes from the National Health and Medical Research Council, which funds medical research, a very broad base of types of medical research. Because we work on muscle diseases, we can also apply to different foundations that specifically fund muscle-related research, such as the Muscular Dystrophy Association of the United States of America and the Association Française contre la Myopathy, which is the AFM, or the equivalent of the Muscular Dystrophy Association in France. We also have funding from other types of patient groups as well, such as the Foundation Building Strength for nemolymopathy, and nemolymopathy is one of the floppy baby diseases that we work on. So it's a mix between funding from the Australian government, which ultimately comes from the Australian taxpayer, and also from funding that's been raised by patients and their families who have got a particular interest in types of muscle diseases.
How did it feel to win the Young Tall Poppy Award in 2010?
It was a great honour to win the first Young Tall Poppy Award for Western Australia. And the Young Tall Poppy campaign recognises people who have done excellent science, but also have a passion for communicating that science. So people who like to talk about what their science is to the general public and to patient groups and to students and all different areas, and participating in encouraging people to think about science. So it was a great award to receive, and I'm still involved with the campaign. It's wonderful to see all the recipients of the years after me and all the wonderful science that they're doing.